I would like to welcome you to this session, Graphene in Nanomedicine. My name is Peter Wick. I'm heading a research lab at EMPA, and it's my great pleasure to chair this session, Graphene in Nanomedicine. In particular, it's a great pleasure because we have two excellent speakers which were very active in the European flagship project Graphene, which is a 10 years initiative, started exactly 10 years ago, and we completed this project by end of September this year. And therefore, it's a great pleasure to have the two speakers here, which can now conclude more or less 10 years of research. So the first speaker will be Professor Dr. Bengt Fadeel. He's from the Division of Molecular Toxicology, Institute of Environmental Medicine from Karolinska Institute. I'm very happy, Bengt, that you're here, and give you the floor for addressing the potential safety aspect and how our immune system is recognizing nanoparticle, in particular the graphene. Thank you for coming.
Okay, thank you so much. This is a short talk, and I'm certainly not going to give an overview of 10 years of research in the graphene flagship. Instead, I will just give some highlights from our latest study. And I will focus, as you can see from this updated title, on graphene oxide, which is one of the most studied members of the graphene-based family. And I will discuss immunological interactions, but actually, while we have been studying quite a lot of effects of graphene oxide and other graphene-related materials on immune cells, what I will discuss today are the indirect implications or interactions with the immune system and the role of the microbiome, which I think is so far neglected in this research field. So first, a bit of background on the AH receptor, or the aryl hydrocarbon receptor. For those of you who might be students of toxicology, you may remember this as a dioxin receptor from your old textbooks. But it's not only, or maybe not even mainly, a dioxin receptor. It is, in fact, a sort of environmental sensor that responds to a variety of different ligands, both endogenous ligands, dietary ligands, and ligands generated by the gut microbiome from the diet. And as is mentioned in this slide, I guess we have no pointer here, but the AH receptor is emerging as a regulator of immune responses, both innate and adaptive immune responses. In particular, and as is illustrated here, in the immune responses in the gastrointestinal tract. What you see here, actually, in the schematic on the right-hand side, is a family of dietary metabolites, all generated from tryptophan, which in turn affect the AH receptor, which is a transcription factor, which then drives gene expression and regulates a number of physiological processes in the body. I will discuss another class of AHR ligands today, which are the short-chain fatty acids. And we have been using the zebrafish model as our model. So GO in the title of this slide stands for graphene oxide, whereas BA stands for butyric acid or butyrate. Butyric acid is one of the short-chain fatty acids that is produced by microorganisms, bacteria, in our gut. And there's some evidence that butyrate actually acts as a ligand for the AH receptor. And we do, in fact, confirm this in our study. So if we start with the data on the left-hand side, we exposed zebrafish embryos to graphene oxide, and we performed, sectioned these specimens and performed transmission electron microscopy. Of course, this is pretty much looking for a needle in a haystack, but we do see graphene oxide sheets. But to really verify this, we then did Raman confocal mapping of the whole embryos, and we can detect the characteristic Raman spectra with the D bands and the G bands and so forth of graphene oxide in situ in the gut of the zebrafish embryo. So, having established that graphene oxide is internalized by these organisms, we then performed the exposure either in wild-type zebrafish or in AHR null zebrafish, which the AHR gene in zebrafish is called AHR2. So AHR2 null versus wild-type. And we looked here for an indicator, or let's say a biomarker of AHR activation, which is CYP1A induction. CYP1A is one of the most important downstream genes of the AH receptor. What you can see here, if you look closely, otherwise I refer you to the paper, is that graphene oxide alone has a very minor effect, and butyrate also a very minor effect in this model on CYP1A expression. But if we combine the two and then expose the zebrafish to the mixture of graphene oxide plus the short-chain fatty acid, which is butyrate, we see a strong induction of CYP1A. And this is abolished. If you go to the bottom right-hand corner of this slide, this is abolished in the AHR null zebrafish. So we have proof that this is an AHR-dependent induction of CYP1A. What is also interesting, if you compare the top row, the graph to the right versus the graph to the left, we only see this effect in germ-free zebrafish embryos. That is, the effect is revealed when the zebrafish embryos are rendered germ-free. We don't see it in the conventional, which is CV, as you can see on the left-hand side. We also wanted to know, so this is of course PCR done on whole zebrafish embryos. We wanted to know if this CYP1A induction occurred in the guts, and to this end we used the CYP1A transgenic zebrafish strain, which is CYP1A EGFP transgenic. And we do see, after exposure to the combination of GO plus butyrate, that the CYP1A lights up in the gastrointestinal epithelial cells. In this experiment, we also used fluorescent butyrate, so we see the butyrate in the gut lumen, as you can see in the images at the bottom of this slide. We then asked whether immune-related genes were affected, and we performed PCR for a range of genes. I'm showing you here on the left-hand side of this slide the LCK, which is an important lymphoid marker. So these are PCR data on the left-hand side, and again in the germ-free background, the combination of GO plus the short-chain fatty acid derived from the gut microbiome triggers a very strong induction of this lymphoid marker. This is completely abolished in the AHR null, which you can see in the bottom left-hand corner of this slide. So, and again, we use another transgenic line. This is now the LCK EGFP transgenic line. And CV, I remind you, stands for conventional, and GF stands for germ-free. So the top part of this figure shows no treatment. We do see some LCK signal lighting up in the thymus. This is expected. This is a lymphoid marker. But if you go to the bottom row of the CV and GF samples, what we see is actually a homing of LCK positive cells to the gut. This is just a representative image, but we have actually quantified this. So there is a strong induction of LCK, which is a lymphoid marker, and homing of those LCK positive cells to the gut. Now keep in mind that these zebrafish are only 5 days old, so there is not really a fully developed adaptive immune system. So what are these cells that are positive for a lymphoid marker, if not T cells? So at this stage in the development of the zebrafish, there are no fully developed B cells and T cells. We reasoned that this could be a marker of innate lymphoid cells, and in order to actually demonstrate this, we performed single-cell RNA sequencing of whole zebrafish embryos that had been rendered germ-free and were either not exposed, these are the control samples, or the control sample to the left, versus the zebrafish exposed to GO, our graphene oxide, plus butyrate, which is the microbial metabolite or short-chain fatty acid. I don't know how familiar you are with single-cell RNA sequencing data, but this is essentially what you get, what the software, in fact, the statistical method allows you to display the data as clusters. Now, each cluster represents a cell type. based on its gene expression profile. And each dot is a cell. So this is single cell RNA sequencing. What we find is that there is a population shown in green in the left-hand side of this slide of LCK-positive lymphocytes or T cells. This is not unexpected. There is some emergence of cells from the thymus at this stage. But after treatment to graphene oxide plus butyrate in a germ-free background, what we see is the emergence of another population, which are LCK-positive cells, but without the T cell receptor. So they cannot be T cells. And therefore, by definition or by inference, we believe these are ILC-like cells. Now, ILC stands for innate lymphoid cell. But all the other populations or clusters in this diagram are other cell types, gastrointestinal cells, neurons, and so forth. And we wanted to go one step further to refine our analysis. But having said that, what we can conclude at this stage is that it's very likely that there are innate lymphoid cells in the zebrafish. There's only one previous study demonstrating the existence of such cells in zebrafish. This is the reference at the bottom of this slide. But in that study, the authors used adult zebrafish which were RAG deficient. So in order to exclude any involvement of the adaptive immune system. Now, as I said, we wanted to go one step further, so we resorted again to the LCK EGFP strain. Now, this is and we repeated the exposure to graphene oxide plus butyrate. We first made this zebrafish germ-free, as I said, because none of these effects are really revealed in the conventional background. What does it mean that we used LCK transgenic strain? Well, we exposed the whole zebrafish, obviously, but we then dissociated the zebrafish embryo and sorted on live LCK-positive cells. And we then performed single-cell RNA sequencing only on LCK-positive cells. So in this way, refining the study, because every cluster in this diagram that you see on the left-hand side now represents LCK-positive cells. All other cell types are excluded, and we can do a much more refined annotation of the different clusters. And again, we do see upper left-hand corner of the graph for control individuals that there are some ILC-like cells, even in control. But then, if you go to the lower diagram, and the lower right-hand corner of that diagram, we were able to identify, based on gene expression, which is shown to the right, certain populations which are ILC2 and ILC3-like cells, mainly ILC2. And again, I would have to refer you to the publication, because these dots in the diagram on the right-hand side are certainly very small and it's hard to see. But what we have found, each dot here, by the way, in the right-hand diagram represents expression of a certain gene, and the size of the circle is the percentage of cells expressing that gene. What we see for the first time, actually, is the existence, or what we could demonstrate, is the existence of innate lymphoid cells, which also have markers of regulatory cells. Normally, FOXP3 and IL-10 are expressed in regulatory T cells, and this sort of regulatory innate lymphoid cell has remained elusive. No one has actually been able to identify this in humans or in mice, but we show in zebrafish exposed to graphene oxide plus butyrate that there is an induction of regulatory innate lymphoid cells. Okay, I'm going to wrap up here, and basically what I wanted to show you is that 2D materials, in this case graphene oxide, can certainly affect immune cells directly, but we didn't discuss that today. What I showed you here is that they can indirectly affect immune responses, in this case, immune responses in the gut. And that this occurs through interactions with the microbiome, or to be more specific, through interactions with metabolites produced by the microbiome, which are the short-chain fatty acids. Specifically, using single-cell RNA sequencing, we could show that if you expose these zebrafish to GO with a sort of, let's say, corona of short-chain fatty acids, this induces an ILC2-like response in zebrafish. And we also show that this occurs through the AH receptor, since we found no such response in the HR null zebrafish. So in fact, our model here is that when the zebrafish are exposed to graphene oxide plus butyrate, this affects the AH receptor in gastrointestinal epithelial cells, not in immune cells, in epithelial cells, which in turn produce a signal. We don't know what that signal is, but this signal induces the induction and recruitment of immune cells to the gut. And these are the innate lymphoid cell type 2 like cells with attributes of regulatory cells. Now, what is this innate lymphoid cell type 2 doing? It is typically a cell that is induced, and an immune response that is induced in response to parasites or parasitic infections. But also, these so-called type 2 responses also play a role in allergic and diseases and asthma. We also showed in this study, which I didn't show you, but that the GO itself affects the gut microbiome composition in adult zebrafish. So that was also part of the story. Now, why am I telling you this at a clinical nanomedicine meeting? Well, if we want to use 2D materials as excipients, dare I use this word, or carriers of drugs or other let's say pharmacological entities, we have to consider that the 2D materials themselves may have an effect, as I put here, for better or for worse. I mean, if it's unexpected, it may be for worse, but if we learn how to modulate the immune response using 2D materials, we may actually be moving from biologicals to nanologicals, that is, nanomaterials that are active ingredients in and of themselves. We can discuss this later if you wish, but this is what I think is the message here for the nanomedicine community. Now, none of this would have been possible or even remotely possible without the work of this outstanding postdoc, Gu Tao Peng. She did all the work, and she now has a faculty position in Shanghai. We are also indebted to Harry and Hanna, who helped us with the 16S RNA sequencing, which I didn't show you, and Kostas and Neos, who helped us with the characterization of the graphene oxide, which was, however, sourced from a company, from the company Graphenia, also a partner of the Graphene Flagship. All of this work was funded by the Graphene Flagship. And since I mentioned graphene and beyond in the abstract, I refer you to the reference at the bottom of this slide, which is a review on graphene and beyond, and the interactions of such materials with the immune system. Thank you.